Guys, I don't know, flea beetles may not be hanging today.
What's your favorite part of the research when you're outside?
Finding things that I don't know what they are. just inspiring my curiosity further. Which is, it's more than just inspiring, enjoyment and curiosity. That's how you really start asking the good questions, the meaningful questions, in my opinion, the meaningful questions. By spending a lot of time looking at things and having enough of a basis in insect taxonomy or plant taxonomy, some background in whatever field of science you want, to be able to generate a question. You see, that's pretty strange what I'm seeing there. Like, what's going on with that?
And how did you gravitate towards insects?
I gravitated towards insects because when I spent my first field season in Ecuador, the plant diversity in this cloud forest in Ecuador was just phenomenal. The beta diversity and turnover in plant community over minute spaces of time blew my mind, and I got to thinking about how would the insects be correlating, predictive of this change in plant diversity. Are we going to get simultaneous changes in insects along with it? Do the insects provide enough of a selective pressure to keep certain species that you find 100 meters up from existing here because herbivory pressure would be intense? Perhaps because there's been a competitive release, or there's no predators there in that spot, or something like this. So the insects, at first, were more of a tool, a lens, so to speak, to be able to look into how the greater ecological community could persist, maintain, assemble in the ways that it does. And then after seeing enough insects, and starting to learn them and learn more about the intricacies of their life, I realized that this is just a fantastic way to dive into the natural world, because they interact with so many other things in their environment. Okay.
I am going to continue.
Not seeing any flea beetles today. And what I'll do now is I'll sample 3 leaves from this plant, all about the same age, more or less, and I'll bring those leaves back to the lab this afternoon. I have a meter that'll measure the parts per million of hydrogen cyanide that are present in the leaves. Take a mortar and pestle, grind it up, put it into this little closed vacuum apparatus, and look at the variation in leaf chemistry between maybe this individual Passiflora biflora, another one up the road 200 meters, or even between the leaves themselves, between branches of the same plant. What I'm finding with all this so far is that individual chemical profiles will be relatively consistent within themselves. Between individuals, with some of these species of Passiflora, it's remarkable, the differences in parts per million hydrogen cyanide, like on orders of magnitude. And given this variation, this heterogeneity in which plants seem to be colonized by these different herbivores that are all quite capable of dispersing and moving around, it's a pretty nice correlation that the chemical profiles are what is governing the ability for particular species to colonize one plant versus the other.